Hello, budding biologists, and welcome back to your video lesson. Our focus today is on DNA structure and DNA replication, and we have a lot to cover. Let's make it make sense. By the end of this video, you should be able to explain that the order of the bases determines the trait of an organism, the phrases double helix and antiparallel, as well as identify components of DNA, including deoxyribose sugar, phosphate, our nitrogen bases, cytosine, guanine, adenine, thymine, Chargaff's rule, and how to calculate the role of hydrogen bonds in creating our sugar phosphate backbone, and understand the function of DNA. Let's start with the basics. DNA stands for deoxyribonucleic acid. It's the molecular blueprint that guides an organism's development and functioning. Remember, DNA is housed in the nucleus for eukaryotic cells, but in the cytoplasm for prokaryotic cells. We often refer to DNA as the universal code for all living things. This statement emphasizes that the structure and sequence of DNA, with those four nitrogen bases, adenine, thymine, cytosine, and guanine, serve as a common language or code that dictates the characteristics, traits, and functions of all living organisms. We're all made up of those same four nitrogen bases. Briefly going through this, we're going to start here at our largest form, the chromosome. Each chromosome is made up of chromatids, the two identical halves of a chromosome. Zooming in on our chromosome, we can see that we have chromatin. This is the relaxed, unwound form of DNA. Zooming in further, histones are protein molecules around which DNA coils. So DNA wraps around these histones to form nucleosomes, which are the basic structural unit of our chromatin over here. Zooming in further, we have the double helix. This is the overall three-dimensional structure of DNA. That double helix is composed of nucleotides, which we can see in the middle here. And as you learned in a previous unit, these are our building blocks of DNA. Each nucleotide, as we zoom in further, consists of a phosphate group, deoxyribose sugar, and a nitrogenous base. Let's be more specific though. Starting back on our chromosome. A chromosome is a thread-like organized structure made of DNA and proteins found in the nucleus of a cell. It contains genes, which are segments of DNA that encode specific traits and functions, and chromosomes become visible under a microscope during cell division. Each chromosome is composed of two chromatids. A chromatid is one of the two identical halves of a replicated chromosome. After DNA replication, during the S phase of the cell cycle, this is the form that we would see each chromosome in. It would consist of two chromatids connected at a central point called the centromere. So this view, what we typically think of as a chromosome, is actually after replication has already occurred. Prior to replication, chromosomes are individual. Over here, you can see an individual chromosome. So this is one chromosome prior to replication. After replication, we have that duplicated chromosome with the two sister chromatids attached at the centromere. Where it gets tricky, we will refer to both of these as a chromosome. So we have a single chromosome, and then we have the duplicated version of that same chromosome. Elaborating on chromosomes, there are a few important things to note. Organisms have two types of cells: somatic cells, which is what we call all of the body cells, such as blood cells, skin cells, neurons, muscle cells, etc., and then gametes, which are the sex cells, either sperm or eggs. Each somatic cell contains two complete sets of chromosomes. One set is inherited from the organism's mother, and the other set is inherited from the father. This pairing of chromosomes results in the cell being what we call diploid, denoted as 2N, where N represents the number of unique chromosomes in a single set. For example, humans are a diploid. We have 23 unique chromosomes. So 2, one from mom and one from dad, times 23 unique chromosomes equals 46 total chromosomes. And that consists of 23 pairs of chromosomes where one is from mom and one is from dad. All right, let's shift over to chromatin. Again, chromatin is made up of long thread-like structures of DNA wrapped around histone proteins. This DNA protein complex forms those nucleosomes, which are the basic repeating units of chromatin. Essentially, chromatin is just DNA in its relaxed form. Write that down. It's DNA in its relaxed form. Instead of being condensed into a chromosome, it typically exists in the state of chromatin. During interphase, the phase when the cell is not actively dividing, it's important for cell processes such as DNA replication or transcription to have easy access to genetic material. The DNA, therefore, is in this relaxed state to allow those processes to occur. All right, zooming in from our chromatin, again, we have histones. These are protein molecules around which our DNA coils. Picture it like a spool of thread. This coiling helps in organizing the genetic material, ensuring it's neat and tidy for those cell processes to occur. All right, that brings us to the building blocks of DNA, our nucleotides. Each nucleotide consists of a phosphate group, a deoxyribose sugar, and one of 4 nitrogen bases. The nitrogen bases are adenine, thymine, cytosine, and guanine for DNA. The sequence of nitrogen bases along a DNA or RNA strand carries genetic information for the organism. One additional thing to note, so each nucleotide is made up of a phosphate group, a pentose sugar, specifically deoxyribose sugar, and our nitrogen bases. But complementary nucleotides, so we have a nucleotide here and then a nucleotide here, are connected to each other via hydrogen bonds. That's going to build our ladder-like structure of DNA. Here's the crucial part. Order matters. Please write this down and start in your notes. The specific sequence of nitrogen bases, the order of those nitrogen bases, is what determines the traits and characteristics of an organism, such as your eye color, your hair color, your texture, whether or not you have dimples, etc. Looking closer at the overall shape of DNA, it's said to be in the form of a double helix. Back in 1953, 2 scientists, which you can see pictured here, James Watson and Francis Crick, along with the help of Rosalind Franklin's X-ray diffraction, proposed a model for the structure of DNA. Watson and Crick referred to this structure as a double helix. The term double helix reflects 2 key features of DNA structure. Double, because there are 2 strands of DNA running parallel to each other, and helix, because the structure is helical, meaning it has a spiral or twisted shape. Looking at our image here, you can see that the nitrogen bases sort of resemble the rungs of a ladder, while the sugar and phosphate form the sides. We call that the sugar-phosphate backbone, and together they create this spiral staircase-like structure. And that's what we're referring to as a double helix, is the spiral staircase-like structure. Analyzing that sugar-phosphate backbone closer, we can also see that there are 2 strands running alongside each other, but they actually have opposite directions. We refer to this as anti-parallel. Parallel, meaning they run alongside each other, but anti because of the fact that they actually go in opposite directions. As you can see in our image, one of the strands runs from the 5' end to the 3' end, and the other runs from the 3' end to the 5' end. The numbering of the ends 5' and 3', is based on the carbon atoms in the deoxyribose sugar of each nucleotide. Starting at the tip of that pentagon, the carbons are numbered clockwise. So 1, 2, 3, 4, 5. The 5' end has a phosphate group attached to the 5th carbon of that sugar, and our 3' end has a hydroxyl group attached to the 3rd carbon. These bonds are what help hold that sugar phosphate backbone together. All right, that brings us to one of our most crucial aspects of DNA structure, complementary base pairs. The term complementary base pairs is used to describe these pairings because the bases are like puzzle pieces that only fit together in these specific combinations. Adenine always pairs with thymine, and cytosine always pairs with guanine. This specificity in pairing is due to the hydrogen bonding interactions between the nitrogen bases. If we look over here at our actual images, you can see that adenine and thymine are held together by 2 hydrogen bonds, while guanine and cytosine are held together by 3 hydrogen bonds. Transitioning to the concept of Chargaff's rule. In the late 1940s, Erwin Chargaff, an Austrian biochemist, published an observation describing the specific pairing of nucleotides in DNA. In his publication, Chargaff stated that the amount of adenine equals the amount of thymine, and the amount of cytosine equals the amount of guanine. This concept seems pretty fundamental, as we already know about these pair rules, but this actually has other implications. For instance, if you know the percentage of one base, you can deduce the percentages of the others. Please summarize this in your notes and be prepared to calculate problems like this in class. To calculate, you would follow this basic formula. Let's say you're given the percentage of adenine, A, which we're going to denote as percent of A. According to Chargaff's rule, if A is the percentage of adenine, then thymine should also be approximately equal. So you can estimate that the percentage of the given base is equal to the percentage of its complementary base. From there, you can subtract that combined percentage from 100. So 100% minus that combined percentage would give you the percentage of your leftover bases. These leftover bases are approximately equal to one another. So we can estimate the amount of both by simply dividing that number by 2. Right. Over here, I have an example for you. If you're given adenine as 10%, then based on Chargaff's rule, you know that thymine would also be approximately 10%. Together, adenine and thymine make up 20% of the DNA total. We can subtract that 20% from 100. You only have 100% of something. That's going to give us 80% left over for our other 2 bases, cytosine and guanine. Using Chargaff's rule again, we know that cytosine and guanine are approximately equal to one another. So we can divide by 2 since we have 2 bases, and we can calculate that each cytosine and guanine is approximately 40%. Shifting gears, let's move from DNA structure to that process of DNA replication. Quick reminder, DNA replication is a key event during S phase of interphase. Again, I can't say it enough, star this in your notes. All right, DNA replication is the process by which a cell makes an identical copy of its DNA. This occurs before cell division, ensuring that each daughter cell receives a complete set of genetic information. For replication to occur, the DNA molecule unwinds, and each separated strand serves as a template for the synthesis of a new complementary strand. We call this process I just explained the semi-conservative model. There were actually two proposed models for DNA replication, the conservative and semi-conservative models. In the conservative model of DNA replication, the entire double-stranded DNA molecule serves as a template for the synthesis of an entirely new double-stranded molecule. So as you can see over here, at the end of replication, one of the resulting DNA molecules would consist entirely of the original parental strands, and the other would be an entirely new molecule. Watson and Crick showed that it actually follows our semi-conservative model. So go ahead in your notes. I do want you to know about this, but put a big X through it because we're going to focus on semi-conservative. Essentially, they found that each of the two DNA strands serves as a template for the synthesis of a new complementary strand. After one round of replication, each new DNA double helix consists of one original parental strand and one newly synthesized daughter strand. So in blue, we have our original strand. We're going to call that our parental strand. And in yellow, we have that daughter strand. This model suggests that the genetic material of each DNA molecule is composed of one old strand and one new strand. All right, so again, Watson and Crick showed that it is a semi-conservative model. All right, let's break down exactly how this semi-conservative replication takes place. Keep in mind that DNA replication is a complex and highly regulated process that ensures the accurate duplication of genetic material. All right, replication begins at specific sites on the DNA molecule called origins of replication. And the first step after the process is initiated is to unzip and unwind the DNA. To do this, there's an enzyme called helicase. Helicase breaks those hydrogen bonds between complementary base pairs, creating a Y-shaped structure known as the replication fork. So this right here is our replication fork. As helicase progresses along the DNA, it continuously unwinds the double helix, causing that fork to get larger and larger. Step 2 is actual replication. A different enzyme known as DNA polymerase adds complementary nucleotides to the 3' end of the existing DNA strand. DNA polymerase can only synthesize DNA in the 5' to 3' direction. Star that please, new strands are always built in the 5' to 3' direction as DNA polymerase moves along the template strand in the 3' to 5' direction. As it goes, DNA polymerase adds free-floating nucleotides to create the new strands, and it proofreads or corrects any mistakes that are noticed. We have a few important vocab words for step 2. There are two strands, leading and lagging. Our leading strand is one of the two newly synthesized DNA strands. It's called the leading strand because DNA polymerase can synthesize it continuously in the 5' to 3' direction. So as we find our parent strands, 3' end, so it's going to follow from 3' to 5' on the parent end, but it's actually building 5' to 3'. For this strand, DNA polymerase essentially follows helicase. So here's helicase unwinding and unzipping that DNA, and DNA polymerase is just going to follow it along as helicase unwinds the parent strand. The lagging strand is the complementary DNA strand synthesized in the opposite direction to the movement of the replication fork. So as we can see, helicase is unwinding that way. Our lagging strand is going to be built from the 5' to 3' direction, going away from our replication fork. Due to the antiparallel nature of DNA, the lagging strand must be synthesized discontinuously. It's built in small parts known as Okazaki fragments. Okazaki fragments are short segments of DNA, typically around 100 to 200 nucleotides in length, on the lagging strand. So they're only composed on the lagging strand, and DNA polymerase synthesizes each Okazaki fragment individually, and then they're joined later on. The final piece of our puzzle is ligase. This enzyme acts like glue, binding those Okazaki fragments together. It ensures that the new DNA strands are seamlessly stitched and that there are no gaps between the Okazaki fragments. Replication does continue bidirectionally, going both ways until that entire DNA molecule is replicated. All right, essentially, that is the short version of how replication works. Make sure you know what each of these enzymes does and in which order they occur. So, first, DNA helicase unwinds the DNA and creates a replication fork. Then, DNA polymerase adds free floating nucleotides together. Of course, know that there's a lagging strand and a leading strand. And finally, ligase glues the Okazaki fragments together to create the two new strands. All right, this is a hefty video. Let's wrap it up here. This is a very important part of our next 3 units. So if you're struggling at all, please schedule time at tutoring. Otherwise, I'll see you all in class.